Okay, now we shift from the history of neuroscience to talking about the brain itself. And to begin with, we're going to talk about the individual functional unit of the brain, as Santiago Ramon y Cajal had theorized, that neuron doctrine we discussed. So we will talk about the individual neuron or brain cell, and it has four basic parts to it. It has a soma. Soma means body, so this would be the cell body. Then there's a major fiber running out from the cell. We mentioned that Ross Harrison espoused the outgrowth theory and found that indeed the axon was growing out of the cell body, the soma. On the other side of the soma, we have dendrites. Dendrite means tree, and we can see then why these were called dendrites. And then back to the other side of the neuron, in fact, the far side of the axon, we have the terminal bouton, or terminal buttons. Taking a more three-dimensional-like view of the neuron, we can see here, close to the center, we have the cell body and all its parts inside, including the nucleus. We have the dendrites over on the left side, and we have the axon extending to the right. And the axon is wrapped in myelin sheaths, which we will talk about later in our lectures on the brain. And that axon, if you follow it all the way down, you will see little spiny tips at the end of the axon. These are those terminal buttons we were just referred to. Now, when we say a neuron fires, it fires an action potential down that axon. And when that axon reaches the terminal buttons, it causes the release of neurotransmitter into the synaptic cleft. That neurotransmitter propagates across the synaptic cleft to the dendrites of the next neuron. As we mentioned when we talked about the Golgi staining procedure, the neurons in the brain are kind of a massive tangle of fibers. But what is holding these cell bodies and axons and dendrites in place? Well, these are the astrocytes. Astrocytes are a kind of cell in the brain that not only hold neurons in place, but actually connect the neurons to blood vessels. So if we look over here on the left, we can see a blood vessel, and of course that blood vessel has nutrients to supply to the neurons. How does it get those nutrients to the neurons? By transporting the nutrients through these processes in the arms of the astrocytes. These arms absorb the nutrients and carry them through other arms to where they are connected to the neurons. So not only do the astrocytes serve as a glue holding the neurons in place, but they also help to bring nutrients from the bloodstream to the neurons. And then they even have a third function, which is to create the blood-brain barrier. All of those little arms that are attached to the blood vessels, they're attached more densely than is shown in this image. So densely, in fact, that there's just tiny spaces in between those processes, those arms. And therefore, they cover the blood vessel and allow only relatively small molecules to get through, keeping back more toxic molecules. That is, molecules that can go through the bloodstream and not harm other parts of the body, but that are harmful to the brain. Therefore, these astrocytes form that blood-brain barrier to keep those harmful toxins in the bloodstream away from the brain. Now back to neurons, and there are three basic types of neurons: interneurons, motor neurons, and sensory neurons. Sensory neurons are ones that bring sensory information into the brain. So sensory neurons can be in the skin, they can be in the eyes, in the ears, etc. And they send their signals from the sense apparatus to the brain. On the other end is the output of motor neurons. So these are neurons going from the brain out toward the muscles. And then everything in between sensory neurons and motor neurons is interneurons. aptly named inter, as in between, because these are in between the sensory and the motor neurons. And this is where most of the really interesting psychological stuff happens. The more interneurons an animal has, the more sophisticated of thought processes it's capable of having. So some of your simplest organisms just have sensory neurons and motor neurons with no interneurons. So there's direct connections between the sensory input and the motor output. And as a result, these simpler animals have very few responses they can make. A sensory input leads to a specific motor output. With more sophisticated animals, however, such as mammals, you have lots of interneurons in between. So any given sensory input could lead to a variety of different motor outputs based on all the interneuron processing that happens in between the sensory and the motor neurons. Now let's look closely at the axon. We're going to take a little section of the axon and blow it up here in this image on the left. And what we see on the image on the left are something called microtubules. These make up the cytoskeleton of the neuron. That is the skeleton of the cell. And not only does this cytoskeleton give shape and structure to the neuron, in the axon, these microtubules actually act as a transport system, kind of a railway system, or perhaps more like a sidewalk, because what you get are these pairs of kinesin, just a certain kind of protein that is able to walk along a microtubule, as we see in the GIF image here on the right. It can kind of walk down these microtubules, and on the other end of the kinesin, it can carry what we call vesicles. And vesicles are filled with interesting substances such as neurotransmitter or neurotransmitter precursors. So these kinesin pairs will walk a vesicle full of neurotransmitter from the cell body down the axon to the axon terminal, where it can be released. Now, in Alzheimer's disease, these microtubules tend to break down. This is one of the problems in Alzheimer's disease. These microtubules are held together by a tau protein, T-A-U, tau protein. And these tau proteins hold the microtubules together, and in Alzheimer's disease, it is known that these tau proteins break down, and since they're holding the microtubules together, then the breakdown of tau proteins implies the breakdown of the microtubules. And these tau proteins, when they break down, will form tangles. And these tangles are referred to as neurofibrillary tangles. Now, early research in Alzheimer's disease really focused heavily on these tau proteins and their breakdown as possibly being the cause of Alzheimer's disease. Since then, however, another protein has been honed in on, and this protein is called beta amyloid. And this is a protein that builds up outside of the cell and is a marker of cell death. So the problem in Alzheimer's disease is you get the buildup of this beta amyloid protein outside of neurons that are otherwise healthy. Yet the beta amyloid protein signals that these neurons are dying, even though they're not, and then the immune system will flush those neurons out. Now, one of the first places that this happens in the brain is in the hippocampus, and the hippocampus is a brain region particularly important for memory. So it's no surprise then that Alzheimer's disease involves deficits in memory. So now we've talked a bit about the different types of neurons in the nervous system, and we've talked a little bit about the structure of the typical neuron, having the soma, the axon, the dendrites, and the terminal buttons. But now we're going to shift to talk about the function of neurons. How is it that neurons operate? How do they process information? Well, the first thing we need to know is that there are four important ions that move in and out of the neuron that create its functional properties. So if you're not familiar with ions, ions are charged atoms. Okay, so an atom of any sort is going to have a neutral charge. It's going to have the same number of protons and electrons. Okay, that's when it's an atom. If you add an electron, then you turn this atom into an ion because it now has a charge. It has one extra negatively charged electron, so it's got a net negative charge to it. On the other hand, if you remove an electron, then the atom now has a net positive charge. So again, it's an ion. An ion is any atom with a charge to it, and any atom that has a different number of electrons from protons. So the ions of interest to us in the function of the neuron are sodium, which is abbreviated Na+. So we can see that with this plus sign that sodium has a net positive charge, which means it's actually missing an electron. Same thing with potassium. It has a net positive charge, which means it's missing one of its electrons. Then we'll talk about calcium ions, and there we see a 2+, which means it's actually missing two negatively charged electrons, giving it a net positive charge. And then the only negatively charged ion we'll discuss is chloride. So it actually has an extra electron, giving it a net negative charge. So where are these ions? Well, they're both inside and outside the cell. These ions are held inside or outside the cell by the cell membrane. Cell membrane, as we can see here at the bottom left of the image, the cell membrane is made up of a phospholipid bilayer. Phospholipid bilayer. It's a bilayer because there are two layers, as we see, and it's a phospholipid bilayer because these two layers are made up of phospholipid. And these phospholipids are hydrophilic on one side and hydrophobic on the other side. It's the lipid part that is hydrophobic. As we know, lipid is like fat or like an oil, and oil doesn't mix with water, so it makes sense to think of lipids as hydrophobic, as if they're afraid of water. And it's because of this hydrophobic head or tail, rather, of these phospholipids that water is not able to permeate the neuron. Water cannot move across these phospholipid bilayers. As such, the ions we discussed, the four ions, are dissolved in water. And if the water cannot move across these membranes, then the ions themselves cannot move across these membranes. Yet, we already stated that the function of the neuron is a matter of these ions moving in and out of the cell. But if the cell has an impermeable phospholipid bilayer, impermeable cell membrane, then how are we going to move these ions in and out? Well, there are ion channels inserted into this cell membrane that enable the movement of certain kinds of ions. As we can see here, we see the phospholipid bilayer making up the cell membrane, and inserted into that cell membrane is an ion channel. And the ion channel is just a protein string made up of some polypeptide subunits, and depending on which polypeptide subunits make up that protein string, you get a different kind of channel. So one set of polypeptide subunits will make a protein string that will form a channel that is selective for, say, sodium. That means that it lets sodium go through the channel but no other ion. That is, there's a pore in the center of that channel that is just the right size and shape for sodium, but not the right size and shape for potassium, or the right size and shape for calcium, nor the right size and shape for chloride. Of course, there are also chloride channels that will allow chloride through, but not the other ions. There are calcium channels, etc. Taking a closer look at these ion channels, in this image, we can see running horizontally from left to right, we can see that phospholipid bilayer, the cell membrane. And inserted into that phospholipid bilayer is an ion channel. And we can see the different colors that make up the ion channel. These different colors represent the different polypeptide subunits. Now, in the first row, we're looking at a cross-section of the cell membrane and the ion channels. But if we look at the second row, now we're looking from the top down at the ion channels. If you look over here at E on the second row, you can see that there's really no hole in the center, or if there is a hole, it's a very small hole. If we look over to about B in this image, then we see that a rather large hole has occurred in that ion channel. And that hole is going to be the right size and shape to let one of the kinds of ions through. Well, how do we go from the closed state over here in E to the open state over here in B? We're going to do that by applying electrical charge. The ion channel is normally closed, and if we apply an electrical charge to that ion channel, the protein string will change shape in just the right way to create a pore or a hole right in the center of that ion string that will allow its selected ion to go through. Okay, we've discussed the 4 different types of ions, and we've discussed ion channels and the fact that these ions move through these ion channels. But why do they move through the ion channels? Well, they do so because of 2 factors that influence the movement of these ions. There's the diffusion force and the electrical force. The diffusion force sets up a concentration gradient, and this force or this gradient describes the difference of concentration of a substance. In this case, the difference in concentration of an ion on the outside versus the inside of the cell. If we look over here at the image on the right, we can see the impermeable cell membrane is this line in the middle of the image, and the left side of the image is the outside of the cell, the right side of the image is the inside of the cell. And we can see that sodium and chloride are concentrated outside of the cell. And the diffusion force is trying to push sodium and chloride along its concentration gradient to the inside of the cell, but because the cell membrane is impermeable, it can't do this. So we have some potential energy here. How do we turn this potential energy into kinetic energy? By inserting and opening sodium and chloride channels. So in the second part of this image, B, we see sodium and chloride channels that are open, and we can see sodium is diffusing from the outside of the cell to the insides of the cell, as is chloride diffusing from the outside of the cell to the inside of the cell. And with just the diffusion force alone, we see in part C of the image that there will be equal amounts of sodium on the inside and the outside of the cell, and equal amounts of chloride on the inside and the outside of the cell. However, the diffusion force is not the only one at work. The electrical force is also at work. So, in the case of sodium, sodium is concentrated outside the cell, so its concentration gradient is going to push it to the inside of the cell. And it's also the case that sodium is positively charged and the inside of the cell is negatively charged. Therefore, not only is sodium going to follow its concentration gradient to the inside of the cell, but it's going to follow its voltage gradient to the inside of the cell. So actually more than half of the sodium will go from the outside to the inside of the cell. Chloride's a little bit different. Chloride is concentrated outside the cell, so its concentration gradient is trying to push it to the inside of the cell. However, because chloride is negatively charged and the inside of the cell is negatively charged, The voltage gradient is pushing in the opposite direction, trying to keep chloride outside of the cell. So we note here that concentration and voltage gradients can work in concert, or they can work together, as the case with sodium. They're both pushing sodium to the inside of the cell. Or they can counteract one another, as we see with chloride, where the concentration gradient is pushing chloride into the cell, but the voltage gradient is pushing chloride out of the cell.